Okay, class, we're ready now to talk about structuring of polypeptides and proteins. As I alluded to, proteins and polypeptides have specific shapes. Shape is of a paramount importance with them as far as function. Alterations in shape could denature them and cause them to lose function. Any of the functions we mentioned, contractile, regulatory, enzymatic, whatever. The structuring is in a hierarchical fashion. The first structure is the primary, then the secondary, then the tertiary, and then the quaternary. Okay. The primary structure, which is really the main basis of everything, is what is the linear sequence of amino acids. Which one follows which? Very important, because it's going to determine all the subsequent hierarchical shapes. The primary structure is determined by DNA. So let me make this very clear. The only thing that DNA does directly is to determine the primary structure of a polypeptide or protein. Let me say that again. The only thing that DNA does directly is to determine the primary structure of a polypeptide or protein. So DNA does a lot, but the only thing it does directly is to determine that. Now, why do I use the term directly? Because we are made up of more than proteins. Also, there's other structuring beside the primary. But what we will discover is the primary structure sets the basis of everything. It even sets the basis for the production of enzymes which control carbohydrates and lipids. So you gotta follow the scheme that DNA is doing one thing, but indirectly doing several things. All right. So the primary structure of a polypeptide or protein is the linear sequence, which follows which. Very important. Deviations in that or mutations will change everything. Just like with sickle cell, you can have one mutation out of a bunch of amino acids and it can change everything. So that's the primary structure. Okay. Now we go to the secondary structure. So the primary structure is being held by these peptide bonds. You don't see it here, but go back and study that. Primary structure held by these peptide bonds. We go to the secondary structure. The secondary structure basically is, when you look at the polypeptide or protein, does it have segments that are repeating units? Doesn't have to be the whole thing, but are there segments that have repeats? The two basic types of repeats in segments is something called the beta-pleated sheet or the alpha helix. So along this string, once it folds in the secondary, beta-pleated sheet or alpha helix. Now, the secondary structure, what holds it? Well, the primary structure is like a chain, or a chain of a necklace, pearl beads. It's being held by peptide bonds, covalent bonds. Well, when you fold it, what holds the secondary structure in position so it doesn't just fall out? Hydrogen bonds. Hydrogen bonds among some amino acids, their aura groups, holds that secondary structure. So secondary structure is held. Do all proteins or peptides have secondary? Most do, by far most, but some don't. Now, you got to have a primary. You may not have a secondary. If you don't see one, then you don't have one. Okay. We go to the tertiary structure. The tertiary structure is, how does this structure really fold into three-dimensional space? Now, unlike the secondary, where there were two types, alpha helix or beta-pleated sheet, the tertiary structure can be all over the board. So there are many different tertiary structures. It's whatever that polypeptide or protein must have. Now, let me kind of explain this. In insulin, let's take human insulin. It's a protein. The fold of human insulin is the same in you and me on the tertiary structure. So I'm not saying it's arbitrary. I'm saying that if you compare insulin to collagen to any of the proteins, whatever they're supposed to be, they will be. But it's not like collagen this could be that and then vice versa. All right. What holds the tertiary structure? All the bonds that we ever taught. In some cases, hydrogen. In some cases, disulfide bridges. In some cases, hydrophobic, hydrophilic. In some cases, but again, it has specificity. So if right there, let's say this was insulin again, that there's supposed to be an ionic bond there, it's going to be there in every human insulin that's functional insulin. So what have we discovered? A primary, all must have it. Secondary, most by far will have it. Tertiary, all must have it. Now we go to the quaternary structuring. The quaternary structuring is if the protein is made up of more than one polypeptide. Okay, let's talk about that. Insulin is made up of two polypeptides, an alpha and a beta, let's say. Hemoglobin in the red blood cell is made up of 4. So in one hemoglobin molecule, the globin, which is the protein, has 4 polypeptides in one molecule. They're bonded together. And they all together form a quaternary. So what is a quaternary? I would take several of these single polypeptides, bond them together, and then what is the structure of them all? Each would have a tertiary, but what is the overall structure of the whole entity? Because the whole entity now becomes the one protein. So quaternary structuring will only occur if you have more than one polypeptide. Okay, we go further. So here is something you can look at. Primary, linear sequence. Secondary, beta-pleated sheet or alpha helix. Tertiary, all over the board. Many different forms. And quaternary, you must have more than one polypeptide. What helps the protein fold into shape? One thing is, number one, remember I told you the primary structure determines everything. So the primary structure determines whether there'll be a secondary. It will determine what the tertiary will be. That's the primary. It's the linear sequence because of how the bonding angles would be. But in order to help it fold into shape after a protein or polypeptide is made, you have other proteins, which are helper proteins, we'll say. And these proteins have been called chaperones. Chaperone proteins help it fold into place. So here's a very wealthy individual. He has a butler. Now he's putting his suit on. Okay, the person is. But to put his coat on real well, even though he could do it himself, to put his coat on, the butler assists him in putting it on. The butler is a chaperone to assist him in doing what he's intended to do anyway. So that's what we call the chaperone proteins that you can just glance at. Okay. Now, as I mentioned, if you bend a protein or polypeptide out of shape, it loses its function. And that is called to denature it. To denature it. What bends it out of shape? This is where homeostasis comes in. Homeostasis tries to maintain a constant internal environment in the face of a changing external environment. One of the major features of homeostasis is to try to control the pH of surrounding proteins, the pH. You want to keep a good ambiance for the protein. Now, different proteins like different things. Some proteins like to be in an acidic environment. You have to, like the ones in the stomach. So you have to keep them acidic. Some proteins like to be in a basic environment. Then in those areas, you keep the environment basic. But if the pH gets wrong, or the temperature, or something called osmotic pressure, it will cause it to bend out of shape. Okay? So that's denaturation, right? Here's some general types of proteins. The fibrous proteins, like a fiber, generally are more alpha helixy. The more globular types are generally more beta pleated sheet. And you can see that when you just think about the structure, beta pleated sheet versus a fibrous type of alpha helix. Okay. So here would be collagen right there. There's collagen. Kind of that type of structuring there. And here's hemoglobin. All right. Our next discussion, and I'm going to start a new video for this one, are enzymes. Enzymes are one function of proteins, and I would say it is the major function that proteins have, because enzymes govern the direction of biochemical reactions. So we'll talk about that in our next